I am going to talk about mRNA decay, mRNA turnover, and in particular a pathway called nonsense-mediated mRNA decay and its relation to human disease. This pathway is abbreviated NMD. And I want to start out simply by reviewing what we know about translation termination. So I think you all recognize the codons in red to be nonsense codons. These codons generally don't encode any amino acid, and they signal the release of the nascent protein and ultimately dissociation of the two ribosomal subunits from the mRNA template. So actually, we became interested in translation termination by studying a number of hemolytic anemias. And what we found is that a number of these anemias, and it turns out many, many diseases, are due to frame shift or nonsense mutations that result in the premature termination of mRNA translation. And the patients are sick because they do not have a full-length functional protein. They have a truncated protein instead. But what we found is that when translation terminates sufficiently prematurely, then the mRNA will be degraded. And this is because truncated proteins that would derive from the mRNA that prematurely terminates translation can be toxic to cells. They can function in dominant negative ways, for example. So by studying NMD, we uncovered pretty amazing things about RNA metabolism. And just to review what they are, I think you'll all recognize this to be an overview of some of the steps of RNA metabolism. In the case of RNA that derives from a mammalian gene, a human gene that encodes a protein. And these RNA metabolic steps are linked temporally as well as mechanistically. And just to review, you know the DNA is transcribed in a collinear fashion into a precursor mRNA, and we heard that this precursor mRNA is processed both co- and post-transcriptionally. One of the processing reactions removes the introns, that's the splicing reaction. And so mRNA is, in essence, fully formed in the nucleus. It has a cap at the 5' end, a poly-A tail at the 3' end, and it has the exon-exon junctions generated by splicing, at which point the mRNA is exported from the nucleus to the cytoplasm where it's translated, that's where protein synthesis occurs, and degraded. Our studies of NMD uncovered links between pre-mRNA splicing in the nucleus and mRNA translation and degradation in the cytoplasm. This is work that was done by Hervé Lier, who was a postdoc in the lab, working in my lab in collaboration with Melissa Moore, who was at Brandeis at the time. And these links are called exon junction complexes. You also heard about that earlier today, EJCs. They are deposited about 20 to 24 nucleotides upstream of approximately 80% of exon-exon junctions on newly synthesized, spliced mRNAs. And you'll see why they're important for NMD. They stably contain some of the NMD factors that are required for the NMD pathway. And then we also, actually a different postdoc in the lab, Yasu Ishigaki, discovered a new template for protein synthesis that we call the Pioneer Translation Initiation Complex. And you'll see that NMD in mammalian cells largely occurs during a pioneer round of translation. So this is the first round of translation. And if you look at the nature of the cap binding protein, it is CBP80 and CBP20. It's a heterodimer of an 80 kilodalton and a 20 kilodalton protein that typifies the cap of pre-mRNA. And if the mRNA underwent splicing, this pioneer translation initiation complex will also contain the post splicing EJCs. And this is mechanistically, both of those aspects of the newly synthesized mRNA are important for the NMD pathway, as you'll see. By the way, if you have any questions, could you just interrupt and either raise your hand or ask the question without raising your hand, okay? All right, so even though we discovered NMD by studying human diseases, it turns out that NMD functions to downregulate abnormal transcripts that are a consequence of mistakes the cell makes all the time. And therefore, NMD is a type of mRNA quality control. The cell has many ways to prevent mistakes from doing really bad things to cells. In this case, mRNAs are being degraded if they encode a truncated protein. And this is also called an mRNA surveillance mechanism. And when you think about it, it makes sense for cells to eliminate a potentially deleterious mRNA very early in its biogenesis. And that's what happens here in the case of NMD. It's occurring during a pioneer round of translation. And I want to give you one example of what happens when a mRNA escapes the NMD pathway. And the example pertains to RNA that derives from the human beta globin gene. You'll notice translation initiates and terminates in the first and last exon of the gene. Even when nonsense codons are in that last exon and premature, so PTC stands for premature termination codon, there's the normal termination codon, these PTCs do not trigger the decay of beta globin mRNA, and they result in a dominantly inherited form of thalassemia. So you need one defective allele either from the mother or the father, and you have a very bad disease. What happens is that the beta globin mRNA, even though it contains the PTC, has a near normal half-life. It's stable. It's not degraded by NMD. And unlike the full-length beta globin protein, which forms a tetramer with alpha globin protein, forms hemoglobin, carries oxygen in the blood, the truncated beta globin protein can only form a dimer with alpha globin, and this results in ineffective erythropoiesis and the dominantly inherited form of the disease. Well, you can imagine that we were really interested in understanding how a cell differentiates termination codons that do not trigger NMD, like most normal termination codons, and then those that do, like most premature termination codons, but not all, like the example I just gave you doesn't. And so we moved introns around and we inserted premature termination codons throughout mRNAs, and we came up with the 50 to 55 nucleotide rule. This is a generic mammalian mRNA, and the rule says that if there's a PTC more than 50 to 55 nucleotides upstream of the last exon-exon junction, remember, generated by splicing, then there will be NMD. The mRNA will be targeted for decay by NMD, whereas if there is a termination codon in the light blue region, even though it's a premature termination codon, there's no NMD. So you can see why PTCs in the last exon of the beta globin gene, the example I just gave you, they don't trigger NMD according to this rule. And we don't think there's anything special about the last exon-exon junction. We think all junctions can function in this pathway. And just to give you an idea of what we knew about this rule before we knew about the EJC, this rule makes sense because if a ribosome gets in this light blue region here or anywhere in this area, the ribosome will physically remove the exon junction complex that I told you resides about 20 to 25 nucleotides upstream of the junction. Does that make sense? You'll see more of that. This rule predicts that if an mRNA has a PTC and it doesn't undergo splicing, it will not undergo NMD, and that is correct. Okay, so now that we know the rule, I want to go back to this idea that NMD protects cells from the production of truncated proteins that derive from mRNAs that prematurely terminate translation. So even when disease results from NMD-induced protein deficiency, so the absence of a full-length protein, the disease phenotype may be milder than and different from that caused if the protein, truncated protein, were to be expressed. So NMD protects many heterozygous carriers of PTC-containing genes from manifesting a disease phenotype that would result if there was truncated protein being produced. This is just a really short list of many, many diseases where the phenotype is much more severe with a PTC located 3' to the NMD boundary. And I gave you the example about the thalassemias, where if you have a PTC 5' to the NMD boundary, in the area where the PTC will trigger NMD, you have a recessively inherited form of the disease. But if you have a PTC 3' to the boundary, a dominantly inherited form of a disease. All right, actually, the most abundant target of NMD in cells, they derive from alternative splicing. A third of all alternatively spliced mRNAs in cells are NMD targets. So we have 20,000 protein encoding genes, and we need many more than that number to have the diversity in protein that we require developmentally and for the different cell types that we have. And so alternative splicing is used to generate more than one type of protein from one particular gene. What is it? Almost 90% of human genes are thought to encode pre-mRNAs that undergo alternative splicing. So with this flexibility, though, comes mistakes. And NMD exists, was probably evolutionarily selected for to clean up the mistakes. And I want to give you a couple of examples, well, one example of how NMD is linked to alternative splicing. And in this example, the alternative exon is red. And normally, let's say, it's removed so that translation initiates and terminates normally, and there's no NMD. But if that red exon were retained in the mRNA, and if it had a premature termination codon in frame with the initiation codon, then there's likely to be NMD. Of course, more to the 5' end, this PTC occurs, and more likely then there would be an exon-exon junction and therefore an EJC downstream, and more likely then the mRNA would be targeted for NMD. And another way that NMD could be triggered by retaining this red exon is if it didn't consist of a multiple of 3 nucleotides. And when it's in the coding region, of course, you'll get a shift in the translational reading frame and the generation of a termination codon downstream of the shift. Does everybody get that? So this I'm sorry? Go over it again? Okay, so there are, in this example, here's the alternative exon, but 1, 2, 3 exons, and here's the alternative 4th exon. And let's say retaining that exon is a mistake. Well, what happens when that exon is retained, you can get NMD by 2 different processes, or if 2 different things happen. One is if that red exon had one of the 3 termination codons I mentioned in the first slide in frame with the initiation codon. So the ribosome will start here and end here, truncated protein. But look, there's a exon-exon junction, and you'll see in later slides there'll be an EJC here. And if this distance between the PTC and the junction is more than 50 to 55 nucleotides, that EJC will not be removed by the terminating ribosome, NMD. You'll see more about the mechanism in later slides. And then another way by which you can get NMD, the introduction of a premature termination codon because of splice site choice, is if that exon were not a multiple of 3 nucleotides. So there's the triplet code, and as soon as you get off the triplet code, you'll start reading into a different one of the other 2 reading frames that will most likely generate a premature termination codon and a target for NMD. And actually, this kind of red exon inclusion is a type of intron inclusion, because you can think about this whole thing as being an intron. And other ways to generate an NMD target is by skipping an exon. So if this exon were skipped, for example, it's supposed to be in there and it's not a multiple of 3 nucleotides, you get a shift in the reading frame. And then splicing downstream of the normal termination codon, which is an interesting concept. So computationally, if you look at most normal termination codons, they generally are not followed by a 3' untranslated region that has a splicing event, that has an intron that's removed by splicing. However, if there were a splicing event so that an exon-exon junction were generated more than 50 to 55 nucleotides downstream of the normal termination codon, that's all you need for NMD. And this is an interesting regulatory pathway used by cells in the sense that as many as 70% of genes encode RNAs that undergo alternative cleavage and 3' end formation. So in other words, alternative positions where the poly A tail is added. So the longer the 3' UTR, the more likely you're to have an intron, and the more likely, therefore, the normal termination codon will trigger NMD. So NMD is not 100% efficient. It depends. It can regulate, downregulate mRNA levels to maybe 10% of the normal level. So you're still going to get some protein, but at least it reduces the level of the toxic, potentially toxic truncated protein, so it's not as bad for the cell. Okay? Am I making sense? Okay. All right. And work that we did with Ben Blenko quite some time ago demonstrated that most of the alternatively spliced mRNAs that are NMD targets it's junk because of the mistakes that are routinely made during splicing. And we know this basically for 2 reasons. One is that they're very low in abundance, and most importantly, the hallmark of constructive alternative splicing is that it's taking place in a cell type or developmental stage-specific way. And we don't see either of that in the case of the mRNAs generated by alternative splicing that are NMD targets. All right, I just want to mention that NMD, the cell, of course, is going to do whatever it can to survive as best as it can. And it has usurped this NMD pathway for constructive purposes. And one way it does this is by a pathway called RUST, which is unproductive splicing and translation. It's also called alternative splicing linked to NMD. And it turns out that there are many negative regulatory pathways for splicing factors that utilize the linkage between splice site choice and NMD. And I'll give you an example of that, which is polypyrimidine tract binding protein, PTB. It's a splicing factor. Normally, PTB pre-mRNA contains exon 11. Full-length PTB is produced. But when the intracellular concentrations of PTB become too high, PTB feeds back on the splicing of its own pre-mRNA and results in skipping of exon 11. Exon 11 is in the reading frame. Exon 11 is not a multiple of 3 nucleotides, generates a shift in the reading frame, and therefore a premature termination codon that triggers NMD. So PTB, when its concentration is too high, downregulates its own production by splice site choice that results in NMD. Okay? Okay. Many examples of that. And then what are other natural targets of NMD? mRNAs that have an upstream open reading frame. There are lots of those. We already talked about mRNAs that naturally contain 1 or more introns within their 3' UTR, especially because of alternative 3' end formation. We've done work on selenoprotein mRNAs. So one of the 3 codons that were in red in that first slide was a UGA codon, and usually that doesn't encode any amino acid, but in specialized mRNAs, it encodes the 21st amino acid, selenocysteine. These specialized mRNAs have a selenocysteine insertion element in their 3' untranslated region, which helps the ribosome put selenocysteine in there. And we did studies a long time ago where if you feed a rat more selenium, you get more selenocysteine incorporation, but there's always some level of NMD. So higher levels of selenium, more selenocysteine incorporation, less recognition of the UGA as a termination codon that would trigger NMD. It's always like this, depending upon intracellular selenium concentrations. All right, so I'd like to go over mechanism. Remember we talked about NMD occurring largely during a pioneer round of translation. So here's CBP80 and 20 bound mRNA. Here's the initiation codon with the ADS ribosome already ready to translate. There's the PTC, the premature termination codon. Here's the normal termination codon. And upstream of this exon-exon junction is the EJC. This EJC is deposited 20 to 24 nucleotides upstream of exon-exon junctions. It's a distance-specific deposition. And you'll see that some of the factors, the peripheral factors, are called UPF factors, which we named after their orthologs in yeast. Those are NMD factors, UPF. You don't need to know what it stands for. Those are just know that UPF are NMD factors. So when translation terminates at a PTC, a surf complex forms. This was discovered by Shige Ono's group, a group in Japan. There are the two translation termination factors that are normally recognizing all termination codons. But in addition to those, there's the key NMD factor called UPF1. It's a helicase, an ATP-dependent RNA helicase. And it joins with its kinase called SMUG1 to form surf, taking the first initial of the constituents. Notice how the terminating ribosome has not removed the EJC. That's key. So let's say it removed this EJC, but then there was another one down here. You'd still get NMD. All you need is one downstream. So then, after surf forms, UPF1 and its kinase join the EJC. And then UPF1 becomes phosphorylated by its kinase, at which point there's a really important step of translational repression before there's decay. And we know if you don't have translational repression, you don't get decay. I just want to touch on that concept. So this was work done by Olaf Isken when he was a postdoc in the lab. He talked about how, he wrote about how UPF1 phosphorylation triggers translational repression. So here's UPF1 being phosphorylated by SMUG1 sitting on the EJC. And then what happens is phospho-UPF1 feeds back onto a 43S pre-initiation complex poised at the initiation codon. Actually interacts with the two highest molecular weight subunits of EIF3, which is a multi-protein complex. And then this prevents the 60S ribosomal subunit from joining to form a translationally active ADS ribosome. That's the step of translation repression. And we also know that phospho-UPF1 has a higher affinity than the bulk of cellular UPF1, which is hypophosphorylated. It interacts indirectly with degradative activities that we know function during mRNA decay. And I'm not going to really talk about those, but I can later if you'd like. And they involve decapping activities and 5' to 3' exonucleotic activities, as well as deadenylating activities. This is the exosome that's degrading the body of the transcript in the 3' to 5' direction. And then there's an endonuclease as well. So I think you're kind of getting the idea that we were really surprised that this newly synthesized mRNA and associated proteins, you know, mRNAs and like all RNAs in the cell are always associated with proteins. And those proteins are deposited based on what has happened to the RNA before, in earlier steps of its biogenesis. So before our discovering the pioneer translation initiation complex, only this mRNA and associated proteins was known to support protein synthesis, to support translation. And it turns out there's a precursor-product relationship between the two, and by the time EIF4E, which is the cap-binding protein of the bulk of cellular mRNA in the cytoplasm, before CPP20 and 80 is replaced, by the time CPP80 and 20 is replaced by EIF4E, this mRNP is immune to NMD. And there's two reasons for that. One is, notice there's no EJC anymore, and we know the mechanisms by which it can be removed. And also, CPP80 actually promotes the NMD pathway. And part of that story was done by Jungwook when he was a postdoc in the lab. He described how UPF1 is the key NMD factor. It's the ATP-dependent RNA helicase. It associates transiently with the cap-binding protein of newly synthesized mRNA, and it promotes NMD at 2 steps. So now we're drawing the transient or weak interaction. You can only see this when you cross-link. And we've mapped the interacting regions. That interaction will occur on all RNAs whether or not they're NMD targets. The cell has no way of knowing whether an mRNA should be targeted for NMD or not until it's translated and the ribosome terminates. Where is it terminating relative to the EJCs downstream, if there are any, right? So this is going to occur on all newly synthesized RNAs. And CPP80 actually escorts UPF1 and its kinase to form the SURF complex. We can physically see it escorting. Biochemically, we can see it in immunoprecipitations. And then it physically escorts UPF1 and its kinase to the EJC. So those are at least 2 ways by which CPP80 promotes NMD. And I just want to mention that if we tether, you can experimentally tether UPF1 anywhere on an RNA, and if we experimentally tether UPF1 downstream of a normal termination codon, that recapitulates a splicing event. And by so doing, we override the need for an EJC, because UPF1 is already there. We override the need for CPP80, because UPF1 is already there, and both mRNPs will now be targeted for NMD. All you need is UPF1, and then it will be it's not a very efficient pathway, but it can be stochastically phosphorylated. Okay? Okay, so I just want to end. How many minutes do I have?
Oh, if my watch is correct, lots of minutes.
Lots?
Yeah, because you started at
oh, yeah.
4.
Okay, that's all right. Then we have time for questions and answers. Maybe I'll go slower. Where in the cell does NMD occur? Well, I've told you that NMD targets newly synthesized RNA that maintains the cap-binding protein that's acquired by pre-mRNA, and that maintains the exon junction complexes that's acquired by nuclear mRNA as a consequence of splicing. And so, to the best of our knowledge, this is a translation-dependent mechanism, right? How do you recognize a termination codon? How do you recognize a premature termination codon? Translation. And to the best of our knowledge, translation occurs in the cytoplasm and not in the nucleus, although that's been very controversial. And we knew something that I'm not telling you, and that is we knew NMD targeted mRNA that co-purified with nuclei. And we knew our nuclear pores were intact. We knew even the cytoplasmic filaments from the pore were intact. We made excruciatingly painful experiments to make sure we didn't have any cytoplasmic tags on our nuclei. And so we proposed the idea that NMD was targeting newly synthesized mRNA at a point where the mRNA co-purified with nuclei, but had access to cytoplasmic ribosomes and could be translated. And I'm happy to say, I'll show you some experiments we did in the mid-90s that led us also to believe this. And then I'll show you an experiment we published with Rob Singer last year that nailed it. All right, so how do you measure mRNA half-life? Does anyone here measure mRNA half-life? Okay, so what you want to do is you want to block transcription. So you want to have an mRNA and watch how it degrades. And if you didn't block transcription, you would have more mRNA synthesis, which would confound your measurements of mRNA that pre-existed and then was being degraded. So one way you can block transcription is to drive expression of a gene using the FOS promoter. So the FOS is a gene in cells. It encodes FOS protein is a proto-oncogene protein, and these proto-oncogene proteins have inherently short half-lives because they can transform cells. And they're responsive, their promoters are responsive to serum. So we can drive, for example, the expression of, in this case, we're using triphosphodiisomerase mRNA because there's a hemolytic anemia called triphosphodiisomerase deficiency. TPI is a glycolytic enzyme. So we're driving the normal human gene with the FOS promoter so that that gene would be transcriptionally on when we add serum to serum-deprived cells, and it naturally goes off after about 30 minutes. And how do we know the kinetics of transcriptional induction? We do a nuclear run-on assay. Has anybody done a nuclear run-on assay? Okay. And so the way you do that is you isolate nuclei as a function of time after serum addition to serum-deprived cells. And then when you isolate nuclei, all of the triphosphates go out. They passively diffuse out of the nucleus, and you also lose the energy regenerating system, so you can supplement the nuclei. And whatever polymerase was just pausing while transcribing the gene, when you give it triphosphates, one of which will have an alpha P32 label, will run to the end of the RNA, and you can measure newly synthesized RNA, and therefore look at the transcriptional induction profile. And so here you can see that transcription is maximal about 15 to 30 minutes after the addition of serum-deprived cells, and then it goes down. So because transcription is shut off. And this is true for the endogenous CFOS RNA. But if we drive, in this case, globin is a control, globin gene expression using a metallothionein promoter, it's a promoter that's not responsive to serum, you don't see any decrease in the amount, any significant decrease in the amount of transcription. So first thing we asked is, in the cytoplasm, if we look at the amount of either normal mRNA or mRNA that contains a premature termination codon, we call it ter-mRNA, from a patient with TPI deficiency, what's the half-life like? And we normalized, so we transiently introduced cells with a plasmid that encoded the normal TPI gene. Remember, you have to use the gene because you need splicing. So you get exon junctions and EJCs. So we drove the normal TPI gene with the FOS promoter, and same with the prematurely terminating NMD-targeted mRNA with the FOS promoter. This is in the cytoplasm. Normalize the levels to the level of our reference mRNA. So we also co-introduced a reference plasmid to control for variations in cell transfection and RNA recovery. And if you look at the time after serum to addition to serum-deprived cells, what would you conclude about the half-lives of the two mRNAs? One has a PTC and one doesn't. What are their half-lives relative to one another? Anybody? No one? They're the same, right? Because the rate of decay is the same. You have less ter-mRNA to begin with, but once those RNAs are free in the cytoplasm, their decay rate is the same. So let's see, that's why we say steady-state mRNA, which can be translated, it produces the bulk of proteins in cells, isn't targeted for NMD. NMD is over by the time the mRNP is remodeled to a steady-state RNP. And so we would conclude that the half-life is greater than 12 hours for mRNA, just like it is for norm. What's going on in the nuclear fraction? And so here, it's the same experiment. You see a little bit of loss of normal mRNA because of export, but look at the induction and transcription, because you're starting at a lower level. You see with the PTC-containing RNA, and we know that transcription is pretty much shut off, certainly by 60 minutes, right? Because there's your nuclear run-on experiment. And at 60 minutes, therefore, we can measure mRNA decay, and that's when NMD is occurring. It's occurring on RNA that co-purifies with nuclei. These are free of cytoplasm. It's a hard thing to make, nuclei free of cytoplasmic contamination. That's when NMD is occurring. And I think I've even told you that it's occurring with a half-life of about 15 to 30 minutes in a nucleus-associated way. And by the way, you're all going to get these slides, so you can stare at them later. Fully spliced mRNA is being degraded. Okay. So then, for years, I was asking my friend Rob Singer, can we do experiments where we look at a single RNA? We track a single RNA. Where's NMD occurring when we track a single RNA that has a PTC relative to an mRNA that's normal? And these are really hard experiments. They involve a lot of biophysics. They involve a lot of mathematics. They involve very sophisticated microscopes. And finally, the technology arrived. And we also had a dedicated graduate student in Rob's lab who could work with Hannah, who was a postdoc in my lab and went to Rob's lab to do these experiments. And so this was published last year, characterizing where in the cell NMD occurs. These are not dependent upon the biochemical fractionation studies that I just showed you, because we can look at RNAs directly. And so, a lot of people say, oh, NMD isn't restricted to newly synthesized RNA. And they say that based on half-life studies they do, and they get a curve like this. And they'll say, oh yeah, NMD targets steady-state RNA. And so what we did in this experiment is we did the FOS induction, and at the time that transcription, the burst in transcription was over, we also added actinomycin D, which inhibits like instantly pretty much, further transcription. So we got a really tight block in transcription. And then watched what happened to mRNA that either contained a PTC, the TER, or didn't. And did these experiments, they're called whole cell ensemble experiments, and did a chi-square test and demonstrated, in fact, that the best fit for NMD, the kinetics of NMD, was bimodal, just like I showed you. It's degraded very early in its biogenesis and then escapes the rest of the mRNA. Once it gets remodeled, it has EIF4E at the cap, no EJCs. So it turned out that, according to the mathematical analysis, 60% of the mRNA was degraded with a half-life of about 30 minutes, and the rest had a normal half-life. So that is consistent with what I showed you on the previous slide. And then here comes one of the many experiments that we did. So using intact cells, the cells were transiently transfected with a plasmid that had a bidirectional Pane-inducible promoter. And in one direction, globin, we're back to studying globin mRNA now, that had no PTC, was generated, and in the other direction, globin with the PTC was generated, mRNAs. And so you've got two promoters, and you're generating globin mRNA that has no premature termination codon, and globin mRNA that does. So this is an NMD target. This isn't. It's a rapidly inducible promoter, and notice there's LAC-Z sequences. So this has a sequence from the E. coli beta-galactosidase gene, which will become RNA when the mRNA is made. Here's a different LAC-Z sequence. And so how do you track the RNAs? You take 50 nucleotide antisense sequences that are complementary to these E. coli sequences. We gutted the 3' UTR and put these sequences in it. And these antisense DNA oligos have a fluorescent marker on them, so we can see one of these mRNAs is red and the other is green. And what we found in looking at the ratio of the two mRNAs, so they're produced in the same cell, in the nucleus. So how do we define the nucleus? By DAPI staining. So this stains DNA, and the purple part is the nucleus. Here's the nuclear boundary. Here's the cytoplasm. So if we looked at the number of PTC-containing mRNAs versus norm mRNAs in the nucleus, and we're going the depth of the nucleus, basically that ratio is constant. And then in the cytoplasm, it's pretty much constant, right? So same half-life of norm versus ter. This is lower. Why? Because NMD occurred already. NMD occurred within 430 nanometers of the nuclear envelope, and as far as we can tell, it's on the cytoplasmic side. So that's consistent with NMD targeting newly synthesized RNA. As soon as that RNA is physically accessible to translation by cytoplasmic ribosomes, but still maintains an association with the nucleus. And so Rob did some calculations, and he estimates that the half-life of PTC-containing beta-globin RNA is anywhere from 3 seconds to a minute once it's in the cytoplasm. It's got to get to the cytoplasm. And as we've been saying, its half-life is normal once it escapes NMD and makes it to the cytoplasm free of the nucleus. And this is consistent with translation rates. So it would take about 8 seconds once translation initiated for a ribosome to reach the PTC that comes from this patient. Okay, so I'm almost done. I just want to tell you a few things. So this was Hannah before she went to Rob's lab, and it's kind of cool. She could, so that we have one example of an mRNA that is degraded by NMD in the cytoplasm after it's been released from the nucleus in which it was synthesized. And we could actually augment the time it takes for that mRNA from its time of synthesis to the time it's translated by expressing ASF-SF2. And by so doing, we could move the cellular site of NMD for that RNA, which is normally cytoplasmic to nucleus associated. So it just depends upon how fast does an mRNA, newly synthesized mRNA, associate with ribosomes before it can undergo NMD. And most in the cell undergo that first round while they maintain an association with nuclei. And then, so yes, inefficiently, some mRNAs are 4E bound when they're targeted for NMD. It just depends upon how fast does that initiation of the first run of translation occur relative to when the cap-binding protein 8020, that cap-binding complex of newly synthesized RNA, is replaced by EIF4E. And we've done, Hannah did experiments where she showed that the replacement of the cap-binding protein, CBP80 and 20 by 4E, is independent of translation. And we know the mechanism, and it's published in that paper. So it's a matter of when do things happen on a newly synthesized RNA as to where in the cell NMD occurs and what proteins are bound to it. And I'm done. Oh, so we know a lot about NMD that I'm not telling you, and I'm happy to talk about it. We know about UPF, this ATP-dependent RNA helicase, the key NMD factor, and how it's kind of like molecular gymnastics on the NMD target. And if you want to read a recent review we have on NMD, there's the annual review of genetics. If you don't have access to these papers, just send me an email, and I'm very happy to send them to you. And this is my lab. I kind of acknowledged people who contributed to what I talked about as I went along. And I'm happy to take questions.
Thank you.